right, here we go. In science and health, dun dun dun, scientists analyze 3D model of proteins from disease-causing bacteria. Not just any bacteria. Millions of people are affected by the Streptococcus pneumoniae bacterium, which can cause sinus infections. Yes, I said that live. I have to, middle ear infections and more serious life-threatening diseases like pneumonia, bacteremia, and meningitis. Up to 40% of the population are carriers of this bacterium. Researchers from the University of Victoria used a Canadian light source at the University of Saskatchewan to study proteins that the pathogen uses to break down sugar chains present in human tissue during infections. These proteins are likely tools in the bacterium uses to cause disease. This study further places the new enzyme into the larger context of enzymes that S. pneumoniae, I'm mispronouncing that all the hell and back, deploys during infection. And ultimately, his team wants to know if stopping this initiating enzyme would prevent the next enzyme from causing more damage. Nice thing to find out. The question we are asking partly if the enzyme is able to do this, but mostly how the enzyme acts. The how. You have to figure out the how. It's all about the science, is the how. Says Dr. Boriston, which is why a 3D structural analysis of the protein using data collected at the CMCF was so important. Now that they have analyzed the enzyme, it opens the pathway to developing new inhibitors and eventually possible therapies targeting the enzyme or other enzymes in the cascade may be developed. Our research is thinking ahead to alternative treatment approaches for a bacterium that is constantly adapting to medical interventions. We live in an era where the Streptococcus pneumoniae bacterium is becoming increasingly drug resistant. I'm going to guess here and say it's due to the overuse of antibiotics. I'm sick, taking an antibiotic. I have a virus, taking an antibiotic. I have an infection, here's an antibiotic. Let me see. It is also continuing to change and evade our vaccination strategies, thinking differently and considering alternative approaches to antibiotics. I was right. Is one possible future for fighting infections, said Boriston. When you oversaturate the medical market and give antibiotics for every fucking thing under the sun, you are going to have things that mutate and become resistant to antibiotics. S-I-T-C-I... I can't read. CITES votes to ban trade in endangered otters. Yes, my friends, we must save the otters. That was not clickbait. Over 100 countries voted Sunday to ban the trade in the smooth-coated otter and place it on the CITES most endangered list. The proposal was adopted by 102 votes in favor, 15 against, and 11 abstentions at the Convention on International Trade in Endangered Species meeting in Geneva. Ecologists say this is vital for the survival of the species, which has seen numbers in the wild plummet by at least 30% in the past 30 years, a decline which has accelerated with the fad in Japan of keeping otters as pets. There you have it. Otter cafes have sprung up in Japan, offering baby otters for up to $10,000. The illegal trade has suddenly increased exponentially. Nicole Duplaix, who co-chairs the Otter Specialist Group at the International Union for Conservation of Nature, told AFP this month. The smooth-coated otter and the Asian small-clawed otter are already listed as threatened under the CITES Appendix 2, but India, Nepal, Bangladesh, and the Philippines are asking that they are moved to Appendix 1, which would mean a full international trade ban. So there you go. The otter is endangered. We must save the otter, guys. Save the otter. A microbe that makes methane from oil is found in the Gulf of Mexico. Archaea have been gaining research attention in recent years and occupy their own branch on the Tree of Life. They share some similarities with bacteria. They both like membrane-bound organelles, for example, but their genomes show that they're different. Researchers have now discovered archaea that can exist deep in the ocean and can use hydrocarbons like oil for energy, transforming the chemicals into methane in the process. They fart. They fart. Wait a minute. Never mind. Hydrocarbons can seep out of the seabed, moving through cracks in rocks and sediments from the ground to the surface. These compounds can help sustain a variety of microbial life deep in the ocean. If you guys are wondering why I'm snapping, every once in a while she whines and barks out the window. I have to break her attention. This work, characterized by an archaean called methanoliparia, I had to slow that down on purpose. It can split oils, a long chain of carbon atoms bonded to hydrogen atoms, into methane. It farts. It farts. Sorry, I keep going back to that. I'm a kid when it comes to fart jokes. The reaction is called alkane disproportionation. I practiced that. It was once thought that bacteria and archaea had to work together to make this happen, but this work shows that archaea can do it on their own. This is the first time we get to see a microbe that farts, that has the potential to degrade oil to methane all by itself, said the first author of the study, Rafael Lazo Perez. So it farts. It eats oil and it farts. It has a case of indigestion. I'll continue. I'll stop. Methanoliparia can active the oil. I guess they meant activate the oil and degrade it, creating methane in the process. This article is just a guess. Exactly. A visual observation seemed to confirm this. Microscopy shows that methanoliparia cells attach to oil droplets. Okay, methane is far worse than CO2. We're getting to that. We did not find any hints that it requires bacteria or other archaea as partners, Wegener explained. We scanned DNA libraries and found that methanoliparia is frequently detected in oil reservoirs and only in oil reservoirs all over the oceans. Thus, this organism could be a key agent in the transformation of long-chain hydrocarbons to methane, said Lazo Perez. We have the genomic evidence and pictures about the wide distribution and supposing potential of methanoliparia, but we can't yet grow them in the lab. Why would you want to do that? That will be the next step to take. It will enable us to investigate many more exciting details. Wegener noted, for example, whether it's possible to reverse the process, which would ultimately allow us to transform a greenhouse gas into fuel. There we go. So yeah, they say that it's contributing to the thing. I just didn't highlight it. Here it is. Methane is a major greenhouse gas, 25 times more powerful than carbon dioxide, so organisms that create it could have an impact on our environment if they're widespread. So what they're trying to do is they're trying to isolate these. They're trying to see if they can make them in a lab, and then they're trying to see if they can reverse the process to get rid of methane in the air. In other words, make the microbes unfart and turn it back into oil. Can you unfart a fart? Once you release a fart, it's been farted. There's no way to unfart it. And the next thing we know, we will have a machine in cow asses to generate oil. Yeah. That's coming, Neil. Once they figure this out, that's coming. And that's another video down. If you liked it, there's this. If you didn't like it, there's that. There's a subscribe button if you haven't subscribed yet. If you're not sure, there's more content you can watch before you make that all-powerful decision of clicking the red bell and making it go dark. There's also a bell that's just about as fickle as a pregnant woman in a candy aisle when she's got a craving or she's hungry, and because it only works whenever it feels like it. But the bell's there. In the description below is all the ways that you can support this channel, as well as links to blogs and other channels and so on and so forth. So, until next time, guys, take care of yourselves.